ELISA, short for Enzyme-Linked Immunosorbent Assay, is a popular laboratory test to detect the presence of a ligand in a liquid. How does the ELISA test work? Basically, if we could have an antibody against our target ligand, we could detect the presence of it. Our immune system has the ability to recognize foreign particles and create specific molecules that have affinity to certain parts of it. The foreign particle is called an antigen, and the special parts that antibodies are built against are its epitopes. In running an ELISA test, our job is to follow the traces of our desired ligand, which we are going to call our antigen. But we cannot follow the antigen, we don't know it. What we do know is the structure of our antibodies. So what we do in an ELISA test is that firstly, we represent the antigen that we are following to the immune system of an animal. For small dosages in a rabbit and for larger doses in sheep or a goat. The animal, its immune system is gonna produce the antibody against the antigen, and afterwards we're going to extract the desired antibodies from their serum. In this picture, the circular antigen with 3 different epitopes is our target. As you can see, for each epitope we have a different antibody. The gray parts in our antibodies are specific to certain parts of the antigen, but the blue parts are constant. They are common between different antibodies, and these blue parts are the one that we are going to detect in the next step. So the next step, as you can see in the name, ELISA, enzyme-linked immunosorbent assay, we are going to use an enzyme to detect these blue parts of the antibody. The red shapes here represent enzymes which are going to handle a reaction through which a colorless substrate becomes colorful. In a nutshell, what happens here is that we wanted to calculate the exact quantity of a ligand, or you might want to call it an antigen, in a liquid. To this purpose, we created a specific antibody with affinity to our antigen. For visualizing the complex, the antibody needs to be conjugated with a special enzyme like HRP. The enzyme needs to have the ability of creating color from a colorless substrate. The color afterwards gets detected using spectrophotometry, and you can determine the exact number of your desired ligand by the reports that you get from the device. The test takes place in strips or microplates, usually with 96 wells, with flat bottoms made of polystyrene. It acts as a solid phase on which, depending on your test, you might want to code antigen or antibody for your ELISA test. With this introduction to ELISA, we're going to go through different types of this test with great details. First, we have direct ELISA, which is the base style for ELISA test. As I mentioned before, the reaction happens in the wells of a microplate or strip. In this picture, you can see one well of a microplate, and we are going to follow the reaction step by step. First of all, we are going to immobilize our reagent on flat bottom of the well. So the surface of the plate needs to have affinity to our reagent. The hydrophobic interaction provided by polystyrene or polyvinyl chloride can do the trick. In direct ELISA, the first reagent that gets coated in the bottom of the well is the antigen that we are measuring in the sample. The coating buffer is a physiological saline solution with carbonate bicarbonate with high pH of around 9.6. In the ELISA test, whenever you add a reagent, you need to give it some time for it to find its place in the well. The duration and temperature of these incubation steps could vary in different setups. For example, in one test, an overnight incubation in 4 degrees Celsius might be needed, while in another, incubation at 37 degrees Celsius might be needed for 2 to 4 hours. Alright, now, when the incubation is done, you need to remove the coating buffer by flicking the plate with a rapid movement and then tapping it multiple times on some paper towels. The next step would be to wash the leftovers of the coating buffer using a wash buffer, which usually contains PPS buffer plus a non-ionic detergent like Triton X100 or Twin 20. Depending on your setup, you might want to repeat this step multiple times and optimize the soaking time of each step. As you can see, in the end of coating step, the antigen in your sample gets binded to the surface. Obviously, the more antigen that you have in your liquid, the more of them get the chance of getting binded to the surface of the plate. Of course, some other elements in your sample get binded to the surface of the well as well, but I haven't shown them here in order to not make the picture more messier than it is. You can see that on the surface of the plate, there's still some room which other elements on the following steps could bound to. But we need to prevent them from these non-specific bindings. We have to ensure that all the signals that we get are due to presence of our antigen. So now with a blocking agent, we are going to block these free spaces. Gelatin, casein, bovine serum albumin, and skimmed milk are some protein agents that you could use as blocking agents. So you're gonna fill the wells of your plate with blocking agent and incubate it according to your setup. Afterwards, obviously, the leftovers of blocking should be washed away by filling and removing wash buffer multiple times. Now we have our antigen immobilized on the surface of our well, and all free spaces are blocked too. Now we are going to add the antibody, which is specifically designed by immune system to get attached to our antigen. The antibody is supposed to link the enzyme to the antigen, and because it happens directly, we call it direct ELISA. The antibody could be conjugated with the enzyme, or the two can be marked with avidin and biotin and be added in 2 different steps. As you can see, we've added the conjugated antibody, so the antibody and enzyme are added in 1 step. Now the enzyme has the ability of creating colorful molecules from processing the colorless ones. The most popular enzyme here is HRP, short for horseradish peroxidase, but alkaline phosphatase and beta-galactosidase are some alternatives for it. The small size of HRP speaks in its favor as it takes less room and it is less of a bother for attachment of antibodies. So now we have added the conjugated antibody with enzyme. We need to give it enough time to find its place and bind to our antigen. After the incubation time, we will remove the buffer containing antibody. And in order to get rid of its leftovers, we are going to wash it multiple times using wash buffer. Okay, you're getting closer to the end of direct ELISA. The last step is to visualize the process by providing the enzyme with its substrate. Different enzymes have different and maybe multiple substrates, but if you have used HRP, its most frequently used substrate would be TMB, tetramethylbenzoidine, I guess, from which the enzyme takes 2 electrons and makes 2 water molecules from reduction of hydrogen peroxidases. The oxidized TMB makes a blue molecule which we can determine its amount in the next step. Okay, adding the substrate of enzyme and its incubation time is an important step of your test and must be stopped at the right moment. The stop solution terminates the ongoing reaction by making a drastic drift on pH. Sulfuric acid as a stopper degrades the enzyme and changes the color to yellow. Now we can read the plate using ELISA reader, which uses spectrophotometry. The wavelength is determined by the colored reagents, and the more color it reports indicates more antigen in liquid. Indirect ELISA follows up pretty much the same process as direct one, but the molecule that we are measuring here is an antibody. For example, you want to see if the immune system of the patient has built antibody against a certain pathogen. So let's look at the process in a close-up. Like the direct ELISA, we coat the antigen on the surface, but the difference is that this antigen is not in our sample, and we just use a standard antigen to capture our antibody in the next step. So after washing the leftovers of the antigen, we are going to block the free spaces and wash its remainders. Now we will add the liquid with desired antibody, incubate it for some time, and then wash the plate. For visualizing the captured antibodies or the primary antibodies, a secondary antibody that is conjugated with an enzyme is needed. The secondary antibody has affinity to the primary one and is usually obtained by introducing the extracted antibody from an animal like a rabbit to the immune system of ones from a different species like a goat or a sheep. Okay, we're almost there. After washing the secondary antibody, we are going to provide the enzyme with its substrate and after some time measure the produced color. Again, the more color indicates the more amount of antibody in the tested liquid. The name indirect comes from indirect connection of conjugated antibody with the antigen. But you can see that our target antibody, the green ones here, have an indirect connection with the surface of the plate as well. In one way, direct ELISA could be more precise than indirect one, as you are not coating the unknown serum that contains many ligands aligned with the desired one on the well surface. But on the other hand, cross reactions of two different antibodies in one reaction can sometimes be misleading, which needs to be closely controlled. Competitive ELISA is a style that both direct and indirect ELISA can be fed into. When we don't have access to an antibody for our antigen, like in case of smaller antigens, competitive ELISA is a game changer. In it, a label molecule competes with our desired one. What we do is that we mix our unknown sample with a certain concentration of a labeled molecule. In other versions, the unknown molecule gets loaded to the wells and immediately afterwards its competitor enters. The two types of molecule get attached to the plate's surface, and the one with higher concentration gets the bigger share. After washing the plate, the only remaining step would be to visualize the labeled molecules by introducing their substrate and then measure the produced color. Unlike the previous styles, in competitive ELISA, the more color indicates the less amount of target, as our target molecule competes with the labeled one, and the presence of the labeled molecule actually represents the absence of the desired one. As you can see, the competitive direct style shortens the process and doesn't need a specific antibody, which is good in a way, but as it doesn't use the specific bond between antigen and antibody, the report from basic style is considered to be more precise. I'm not gonna go through competitive indirect ELISA as I'm sure you can guess it already. The last main style of ELISA is sandwich ELISA. The main problem with the previous styles was that the unknown antigen in a serum of multiple other ligands gets coated to the surface of the plate, and in smaller concentrations, the target may not be easily detectable. In sandwich ELISA, instead of coating a serum of multiple components in a well surface, we coat a specific antibody called capture antibody with affinity to our antigen on the surface of the well. To make the coating step more efficient and have the antibodies upright and in correct orientation, some linkers could facilitate the reaction, which I haven't used here. After the incubation time and washing steps, free spots get blocked, the plate will be washed, and the process will actually begin from here. The sample with unknown amount of antigen gets loaded to the well, and now between all its components, only our desired antigen gets attached to the immobilized antibodies. So even the smallest traces of the target would be detectable. Then we would need a second antibody called detection antibody, which is labeled with an enzyme. In this step, you can see the limitation of the process. You can pause the video and guess it. Did you get it? The problem is that sandwich ELISA can only be done with antigens with at least two different epitopes, and we need to have two antibodies completely separated, which might not be very easy. Okay, as always, you're gonna watch the reminders of the detection antibody and introduce the substrate of the enzyme, and finally stop the process by lowering the pH. Once again, here in sandwich ELISA, the more color indicates the more target antigen. In running an ELISA test, the speed of the operation is very crucial as all the samples in different wells need to have the same incubation time. So other than samples and standards, all the other components that are common between different wells like wash buffer, conjugated enzyme, and stopping reagent are poured to a reservoir and being added with multi-channel micropipette. So by accelerating the process, we will make the incubation time of different samples in different wells comparable. Bubbles are another item that could affect the report of ELISA readers, so professional pipetting is crucial to prevent the formation of any bubbles. Another tip for running an ELISA test is that after filling the last well, first set up the timer for incubation time and then apply an adhesive tape over the plate to ensure the liquid wouldn't get vaporized over the incubation step. Okay, let's go through the calibrators and controls of an ELISA test. In all the tests that use spectrophotometry like ELISA test, we need to remove the background signal by detecting the OD of the final buffer as our blank well. So the blank removes the background signal for us. But other than this, we need a negative control. Because of the high sensitivity of ELISA test, it's important to make sure the test components don't have any contaminations to make a false positive result. So we need a negative control well that goes through all the steps of the test except for not having any target reagents. Your test can be done by having a positive and a negative control. That only can report the presence or absence of your desired reagent. But if you want a quantitative report, you need a serial dilution of a known concentration of your desired reagent. These calibrators need to go through all the steps of the test like an unknown sample. In a complicated test like ELISA, with multiple steps and possibilities of operational error, the presence of calibrators is crucial in every run. In the end of our ELISA test, we're gonna have the report of spectrophotometry in our desired wavelength. So by having the exact concentration of each standard sample and the amount of produced color in it, We can have a standard curve by which we can calculate the amount of each sample by its reported optical density that represents the presented color in it. I hope this video was helpful for you. Thank you.